Hi, this is Teemu Arina. I'm the host of the Biohacker podcast and also the curator of Biohacker Summit. And today's guest is well sought after, Dr. Rhonda Patrick, who specializes on aging and cancer and nutrition. And she has done extensive research on those. She's going to be introducing herself in a moment. But she's also going to be speaking at the Biohacker Summit in Helsinki, Finland on the 18th of November. So you may want to check that out. And also, there is another episode that I just recorded with Dr. Tamsin Lewis on biomarkers and how to interpret your blood work. So she's a medical doctor. You may want to check that episode out as well. It's I think it's going to be well linked into some of the things that we are looking in here in terms of inflammation and blood values. But in any case, so welcome to the show, Rhonda.
Thank you. And how did you pronounce your name? Because I completely butchered it when I'm reading it phonetically. I think I called you something that's not actually accurate.
So it's Teemu.
Teemu.
Yeah.
Teemu.
So a lot of people say Teemu.
Yeah.
And it's fine. I'm used to it. It's fine if that goes that way. So Teemu would be the name. And so you are now the co-founder and CEO of Found My Fitness, and you have a lot of YouTube videos on that go actually very deep into genetics and health benefits of nutrients. And you have done extensive research. You have a Ph.D. in biomedical science. So what can you tell us about your background?
Well, my background, so that's correct. I do have a Ph.D. in biomedical science. I started out wanting to be a marine biologist and travel around, on boats and sort of, surf and look at all the marine life. And somehow I ended up, when I was in college at University of California in San Diego, doing chemistry. Somehow I got into chemistry and organic chemistry. And so I majored in chemistry and biochemistry. And I was doing a lot of, synthesizing compounds and drugs and peptides. And I was interested for a while, and I got really bored after a couple of years. While I was still in college, I was doing internships and getting experience, and I got really bored. And so I decided, once I graduated, that I wanted to dive more into biology. So I went and got a job at the Salk Institute for Biological Sciences, which is one of a very, very prestigious research institute in the United States. And I started working in a lab there that was doing research on aging. And they were using these little nematode worms called C. elegans as a research model for aging. These C. elegans have many of the same genes, similar versions of genes that humans have. So it's a really good genetic model to study aging. And I remember the first time I did an experiment where I manipulated and changed one gene, and this one gene had to do with insulin signaling pathway, insulin growth hormone. So it's the same pathway that humans have. And we got rid of that whole pathway in worms. So there was no insulin-like growth hormone, so IGF-1. And the worms went from living 15 days, which is their average lifespan, to 30 days by just getting rid of one gene. So it was a 100% increase in lifespan. And I remember thinking to myself, exactly, wow, like that is amazing that we can genetically change one pathway in this little tiny worm. And that has such a robust effect on its lifespan. And the fact that this genetic pathway was something that was conserved in humans was just even more exciting. So at that point, I thought to myself, okay, I want to do my graduate work in biology. So that sort of, started my interest in aging and biology, and then I became interested in cancer, pediatric cancer. So I went to St. Jude Children's Research Hospital, where I studied cancer metabolism, the role of mitochondria in cancer. So lots of cancer metabolism, and my PhD was in biomedical science more broadly, but specifically my research focused on cancer metabolism.
Right.
So, and then as I was, in graduate school, which was 6 years, I, at the same time, started this blog called Found My Fitness, where I just started putting together little concepts of things I was interested in. I became very interested in, as I was studying cancer, metabolism in cancer, I started to realize the importance of prevention in cancer, because cancer is just such an ugly disease. And so then I started to sort of focus on prevention and nutrition and lifestyle factors, and I put together this blog with my husband. And then I started to pursue that. At the same time, nutrition and, micronutrients, vitamins, minerals, things that are very important for all your enzymes to work properly. Went on to do a postdoc with one of the leaders in the field of micronutrients, Dr. Bruce Ames. And he was my mentor for some time. And at that time, I continued to do Found My Fitness, and even more because he was very supportive of it. And I found that I loved communicating my passion about science, about health and nutrition with people. And so I've just continued doing that with videos I make, as you mentioned, and podcasts and newsletter where I write articles. So it's been a lot of fun. And I'm still a visiting scientist with my mentor, my postgraduate mentor, Dr. Bruce Ames. But I don't, once in a while I go back and I'm doing some experiments, but most of the time now I'm doing what I love doing, and that is just communicating, the health behind, the science behind health and nutrition to people.
Right. Right. So there is so many things there that I want to touch on, from mitochondria to basically things related to longevity and nutrition. I recently watched one of the lectures from Dr. Bruce Ames. It was extremely interesting about the Triad Theory.
Triage Theory.
Triage Theory. I want you to explain a little bit on his work, but he spoke very grandly and respectfully on your contribution to his work in that lecture, actually, which kind of highlights that Dr. Rhonda Patrick is not just any postdoctoral student. And looking at your YouTube channel, it's a wealth of a ton of information on different topics, and you have, as you explained in your background, you have pretty wide kind of outlook and view on human health. Many researchers, they just dive really deep into one aspect of it. You've gone to this kind of looking at these things from very different angles. I would imagine that when you did studies on IGF-1 and then later on cancer, that those kind of growth hormone related things are kind of interlinked, kind of things that make us put more muscle and grow as athletes, for example, can also be deteriorous to our health if getting out of hand. So the time is ticking in a way. So going to those topics, especially your work with Dr. Bruce Ames. So he's one of the pioneers in investigating the effects of micronutrients on human health. So what can you say about kind of the main discoveries that he's done?
I think some of the main discoveries that Bruce has contributed in terms of the effects of vitamins and minerals on preventative medicine has to do with folate. So folate's a very important B vitamin, and Bruce found that so folate is important for a couple of reasons. One, you need it to make precursors to make new DNA. So anytime you have a new cell that's being made, for example, in your gut, in your kidney, in your liver, your brain, anytime you make a new cell, you have to make new DNA, because DNA is the fundamental template of our biology. And in order to make new DNA, you need folate. Anytime you damage your cells, so if you damage, for example, your blood vessel and you're repairing damage, well, you have to make new DNA to make a new cell to repair that damaged cell. So that's a very important biological process, I would say, is making new DNA. And you need folate. Folate's required to make one of the DNA nucleotides, thymidine. So you need that to make it. Without it, you do not make it. And what he found is that when you have very low folate levels, actually levels that are they're equal to what you can find in people that live in the United States that don't eat a very good diet. So a lot of people that are more impoverished They eat a lot of foods that are processed foods that are in boxes or packages. So they're not eating greens. Dark leafy greens is the major source of folate. Spinach, kale, chard, these sorts of vegetables are very high in folate. And so there's a population of people that don't eat, fresh vegetables. They're eating a lot of processed foods. Well,
Right.
he found that very low folate levels actually, because you can't make that certain DNA nucleotide that you need, it actually substitutes a nucleotide that's usually made in RNA, not DNA. So it's not correct. Your body's kind of trying to figure out what to do. What do I do? I need to make this, I need this nucleotide to make new DNA. And so it puts in another nucleotide that's found in RNA called uracil. And what that does is, because that's not really the structure of DNA, that's not how it's supposed to be. It's very unstable, and it causes breaks, single-strand breaks in the DNA. And he compared those single-strand breaks. Now, single-strand breaks are a form of DNA damage. DNA damage is something that's happening all the time. It's a byproduct of normal metabolism. When you're oxidizing, fatty acids, or when you're oxidizing glucose, when you're metabolizing these nutrients that you take in from different food sources, and you breathe in oxygen in order to make energy, that process of making energy also makes byproducts that are very reactive, the reactive oxygen species, and they damage DNA, they damage mitochondria, they damage proteins, tissues, they damage everything. Well, anyways.
Right. So there is a full escalation of all kinds of side effects and issues that arise from being inadequately nourished in terms of food intake, of folate levels, for example. If I understood Bruce Ames' theory also that it sort of like encapsulates also other nutrients in a way that the organism, the human being, for example, is very good at preserving itself in the short term with long-term consequences.
Exactly. Yeah. So, the folate wasn't directly relevant to the triage theory, but he did show that those strand breaks were equivalent to being literally irradiated with X-rays. So it's the same type of damage by not having folate, which really highlights the importance of micronutrients in human health. But to speak more specifically to the triage theory is a theory that Bruce came up with about 10 years ago, that posits throughout human evolution, we as humans have been exposed to periods of food scarcity. And throughout those periods of food scarcity, we also don't get all of our vitamins and minerals, because that's where most of vitamins and minerals come from, is food. And so he posits that our bodies have evolved a mechanism that allows for vitamins and minerals when they're in short, when they're not being provided in abundance, to be triaged to certain physiological processes that are necessary for survival short term, so that you will live long enough to pass on your genes and reproduce and pass on your genes. But those other processes that are not so important for short-term survival right now, but are important for preventing diseases of age like cancer, Alzheimer's disease, those biological processes are not so important, because it doesn't matter if you live to be 80, 88, 90 years old. You just need to live long enough to pass on your genes. That's what biology cares about. So as an example of triage, as you mentioned, Bruce had published a paper showing that vitamin K is a good example, because vitamin K is a very important cofactor for blood clotting. So you need vitamin K in order to have coagulation of your blood, to make sure that anytime you do have, some sort of damage on your blood, your blood clots and you don't hemorrhage and bleed out and die, which is very possible if you can't clot your blood. So vitamin K is very important to that.
That would be unfortunate, yeah.
Yeah, it would be unfortunate, and it certainly would lead to a early death if you were unable to clot blood. However, vitamin K is also very important for activating proteins in the blood vessels that take calcium out of the bloodstream and bring it to your bones. And that's also bring it to your bones and also to your muscle and other tissues that need it.
So is it linked to vitamin D also?
It is linked to vitamin D also, yes. But this function, pulling calcium out of the bloodstream, is only really important to prevent calcium from building up in your blood vessels, which can lead to atherosclerosis. It can lead to vascular dementia. These are diseases that don't show up until you're much older. So he showed that there's a triage where vitamin K, when you deplete a human of vitamin K, actually there's no change in their blood clotting proteins. But there is change in the proteins that usually get activated to pull calcium out don't get the vitamin K. So that's been shown in humans, and then there's been several animal studies that have shown in nice detail that vitamin K, again, when you make an animal deficient in it, it's the blood clotting that gets all the vitamin K and not the other proteins that are involved in pulling calcium out of the bloodstream. So
right on. So I mean, a lot about these things and how they are intertwined and interlinked. So I guess you must have done some dietary changes and you deploy some strategies. I know you're a big fan of smoothies. So, for example, what would be your morning smoothie be like to mitigate potential risk of getting any of that nasty stuff of being malnourished?
Yes, I do try very hard to get a very broad spectrum and large quantity of micronutrients every day, and micronutrients in the form of vitamins and minerals I try to get from a lot of vegetables and plants. So I make sure that I have dark leafy greens. I like to make a smoothie because I've noticed that that I consume more vegetables if I add a smoothie in addition to the salads and steamed vegetables and things like that I also eat. But my smoothie, I have a couple of smoothie videos, and I do a variation of them depending on what I have. But I always have some sort of dark leafy green. I like to have chard, which is high in vitamin K. I like to have kale, which is so chard and kale. They're high in folate, vitamin K. They're high in magnesium. Magnesium is another one that's very important for triage may apply to. Magnesium is essential to make energy. You actually can't make ATP or use ATP without magnesium. So that's something that's essential for short-term survival. But magnesium is also essential for enzymes in your body that are required to repair damage to your DNA and get rid of that damage. Without magnesium, that doesn't work. So magnesium is also very, very high in green vegetables. Chard, kale, and then I like to get my carotenoids. So I like to get the luteins, the zeaxanthin, which are very important for the rods and cones for your eyes so that you don't get macular degeneration. Actually, lutein is very high in kale as well. I'm someone who has higher risk for macular degeneration. So I'm also in that camp of having my sea buckthorn and blueberries and all that and all the dark leafy green vegetables. I mean, some people, especially in the US, you will notice it when you come to Finland, that we are very much into real wild foods, like foraging and collecting your own foods and getting these nutrients from nature. In the US, there is huge focus on supplement this, supplement that, multivitamins and so on. So what do you think? Do we know in science enough yet about all the micro kind of compounds and nutrients that are in foods? Let's take a cup of coffee, for example, has hundreds of different compounds, still some of them unidentified what they do. We have different forms of vitamin C, even in orange, instead of the one form that usually people supplement on. So what's your take on kind of this reductionist approach of taking a supplement? Well, I think you brought up a very important point, and that is that there are literally, hundreds, if not thousands of compounds that are present in, a variety of plants and fruits and vegetables that we have yet to even identify a function for. We don't really know all these compounds, what they are, what they're doing. But as we learn, as technology develops and we have more sensitive assays to measure compounds that are, found in a variety of plants, we start to learn more and more about them. For example, not too long ago, I think it was back in around 1992, a compound was found in cruciferous vegetables like kale or broccoli called sulforaphane. Well, the precursor to it is actually glucoraphanin, but it gets converted into something called sulforaphane, which is a very, very powerful compound that activates this whole genetic system we have called NRF2, which is a very powerful system that activates enzymes involved in detoxifying toxic compounds that we're exposed to, for example, from air pollution, like benzene. Gets rid of benzene. It activates all the glutathione genes, anti-inflammatory. So it's over 200 genes. This pathway is changing. That has to do with detoxification, antioxidants, inflammation, and this is found in cruciferous vegetables like kale and broccoli sprouts, cauliflower, cabbage.
Yeah.
So
one quick question on that. So, to my understanding, the sulforaphane is destroyed by heat. So if I what a lot of people do with kale and cruciferous vegetables is that they steam them or they fry them because of their fear oxalates that you can basically deactivate by heat. So what's your take on that?
Yeah, so again, you bring up a very good point. So these cruciferous vegetables, to make the sulforaphane, so sulforaphane is actually, and I know this is something that we were discussing, it's what I like to call a plant hormetic compound. And what that means is, so hormesis in general refers to a little bit of stress, something that's a little stressful on an animal or on a human physiology, and not too much stress, but a little bit of stress, just enough to activate all these genetic systems that we have that are involved in dealing with stress. So it activates stress response pathways, many of which are antioxidant, anti-inflammatory, things like that are clearing away damaged cells like autophagy. So it's activating all these processes. And what the net effect ends up being is that even though it was a little bit of stress, a little bit of, something that's a little bit toxic to humans, the response to it is so robust that it overcompensates for the little bit of stress, and it ends up having a net positive effect. So sulforaphane is a perfect example of that because in these cruciferous vegetables like kale or broccoli, it's actually, you don't actually find the sulforaphane. It's found as glucoraphanin. Glucoraphanin is the precursor to it. Now, when the plant when the plant tissue is torn, when it's chewed or chopped, for example, if an insect comes and lands on the plant and bites it, what happens now is that glucoraphanin comes in contact with an enzyme called myrosinase. Myrosinase then immediately starts to convert glucoraphanin into sulforaphane, and sulforaphane has an effect on insects that it doesn't kill them, but it basically scares them off. It affects their nervous system. It affects they don't have brains, but they do have neurons. They have a nervous system, and it sort of just confuses them, and so they go away and they don't come back because they remember that plant. So it's a defense mechanism. Well, it turns out in humans, it also is slightly toxic, the sulforaphane.
Right. So, but it's kind of a hormetic stressor.
It's a hormetic stressor. Yeah. But to sort of answer your question, because I went off on a tangent there about heat, it's actually the enzyme myrosinase that's heat sensitive. So you need the myrosinase to be active in order to convert all that precursor that's contained in the plant called glucoraphanin into sulforaphane. And it is heat sensitive. So when you heat up your broccoli or your kale, you end up inactivating a large amount of that enzyme. And so you're actually not even able to you're getting all the other good micronutrients, the vitamins and minerals, but you're not getting as much sulforaphane. So, and there's lots of, it goes into great detail. In fact, I have a video that I'm going to be releasing, a 45-minute video on sulforaphane. I'm going to be releasing next week, so people can
looking forward to that. So, I mean, which one would you then choose? Like risk for kidney stones or just like being able to fight off cancer? So it's kind of, the diet that we eat is kind of paradoxical in the way how it works.
Yeah. And I've yet to see, there's been clinical studies in humans that were given large concentrations of glucoraphanin and some of these things that are, thought to have this anti-nutrient effect, and there's been no effect on thyroid, no effect on kidney, no effect on liver. They've done animal studies in mice where they're feeding them large quantities. There's, again, no effect on liver or kidneys or, so I think that there's important context to consider here. Maybe if
Absolutely.
you're something.
I mean, the human body is full of different pathways, and it's still, the jury is out there in terms of how different compounds, as you get them, these hundreds of different compounds from a specific plant, how they're co-interacting and what the end result might be. So even if you detect in the ingredient something, it doesn't mean that it's going to end up being absorbed in the wrong type of way, right?
Yeah, and I also I tend to agree with you with getting more of your micronutrients and vitamins and minerals from plants and foods, from whole foods, because the vitamins and minerals are in the right ratios. We have all these other compounds, and that doesn't mean there isn't a place for something like a multivitamin. I do think that there have been studies showing, particularly in people that have poor diets, that they can fill micronutrient gaps and they can actually help. I do take a multivitamin just because it's kind of like insurance for me. There's some trace elements, and I found a high quality one that I like, and so I do take that. But I get most of all my my nutrients, my vitamins and minerals are coming from whole foods. I think that's important.
Right on. So I want to also dive into cold and heat and the health effects of that, because I come from Finland where sauna and ice swimming is a big thing. But before that, I want to go back to folate, and I know that you've done some research and studying closely also genetics and how that plays a role in human nutrition. And folate seems to be the one that's brought up in Nutrigenomics often as an example because of the way how it influences MTHFR or something like this, and the fact that some people should not take folate in the folic acid form, but in an activated form because of some genetic issues. So what can you tell us about methylation?
Well, so I did mention the pathway involved in making new DNA. That's very important for folate. The other important pathway that folate plays a role in is precursors for methyl groups. And these methyl groups are used for epigenetic purposes, which basically means they're changing how much a gene is activated or deactivated without actually changing the sequence of DNA, but instead by putting this methyl group, which is just sort of this structure. It's a carbon with 3 hydrogens attached to it, and it sits on top of a DNA region, and it prevents other proteins that come and sit on top of the DNA to activate it. So it can, in many cases, methylation is involved in turning a gene off. So folate plays a very important role in that process. And as you mentioned, this concept of nutrigenomics, which is the interaction between our genes and our diet, and what we're getting from our diet, different micronutrients, macronutrients, things like that. And one of the classic examples of nutrigenomics is the MTHFR. So humans are all different. We have different versions of genes. These are called polymorphisms, or single nucleotide polymorphisms in some cases, where it's just one nucleotide being changed. And it changes when you have one nucleotide being changed in a gene, it can change the function of the gene. And about almost half of the population, at least in the United States, they have a version of the MTHFR gene, which is involved in producing those methyl groups from folate so that it can be used for a variety of epigenetic reasons. They're not able to do it as well. About 10% of the population has a version of it that's so bad that it's almost like the enzyme isn't doing anything. So it's like really, really bad. And that's in about 10% of the population. So there is a bypass for this, and that is actually taking an active form of folate, like you mentioned, called methylfolate, L5 methylfolate, actually. And so basically it's bypassing. Usually what this enzyme does is it converts, a precursor into 5-methylfolate. And because that enzyme can't convert the precursor into 5-methylfolate very well, taking the 5-methylfolate exogenously, so taking a supplement, can bypass that, and then you can then have the 5-methylfolate to be used for epigenetics. So
right. So in people who have kind of malfunctioning methylation, they see a rise in inflammatory markers like homocysteine.
Exactly.
And so if you want to bring that down, you have that elevated, you may want to check your methylation and genetics on that. What other kind of examples comes to your mind in terms of nutrigenomics that has immediate application that we can use today?
Vitamin D, I think that's one of the really, really major ones, that there are very common gene polymorphisms in an enzyme called CYP2R1, which is, so vitamin D actually is a hormone. It's a steroid hormone. It gets, it's not actually, we call it a vitamin, but it's actually a hormone. It's so important that we make it from the sun in our skin. We make vitamin D3 when we're exposed to UVB radiation from the sun. And then that vitamin D3 gets released into the bloodstream, and it gets converted into something called 25-hydroxyvitamin D. And then the 25-hydroxyvitamin D gets converted into the active steroid hormone, which is 1,25-hydroxyvitamin D. So this enzyme I'm talking about, the CYP2R1, converts the 25-hydroxyvitamin D into the it converts the vitamin D3 into the 25-hydroxyvitamin D. So people have a version of this enzyme that does not function well. And so they actually, for example, if they're taking a vitamin D supplement, they will not convert as much of that D3 into the active vitamin D steroid hormone, which is what's doing all the function very well. And so people, for example, that are taking 2,000 IU or 4,000 IU of vitamin D, or I guess in Finland you probably measure it in micrograms. They may think they're getting enough vitamin D, but if they actually get their blood levels measured, they'll find that their blood levels of 25-hydroxyvitamin D, which is what is measured, it's the stable form, are much lower than they probably anticipated. And so I actually know several people with this gene polymorphism, and they have to supplement with a much higher dose of vitamin D3 than they otherwise would.
I'm also one of those people, so I have to take a lot more, like 10,000 IU, to get even the serum levels to where they need to be. And nothing beats like real sun exposure. And fortunately, I don't have methylation problems, but those are the two things: vitamin D absorption and or conversion to useful form, and the other one is just like most of the people in the world, the biggest nutrition deficiency is iron deficiency, and I don't have problems with that actually. My ferritin levels can rise so easy because of genetic issues. So I have to take a look at those things. So I often advise people when they start on supplementing things like vitamin D, or they start supplementing on whatever, iron or folate or magnesium, to check their existing levels, if they and genetics perhaps as well, to combine those both to be able to adjust the right dosage. So how important do you see testing in general?
I think it's critical. I think that getting a blood test to look at your vitamin D levels, looking at your ferritin, your all these, these micronutrients and also other biomarkers are an important way of quantifying changes that we're making in our lifestyle, and
right.
Because not everyone's measured their gene polymorphisms, which you can do with certain companies. At the very least, get a blood test, because there will be a red flag that goes off if you're taking a vitamin D supplement and your levels are still deficient. That's sort of a red flag that you may have one of those gene polymorphisms.
Right. Yeah.
So I think that's extremely important to do. And for me, for the vitamin D, I like to have my blood levels between 40 and 60 nanograms per mil, which I think in Finland would be, you guys measure nanomoles per liter, I think, which would multiply what I just said by 2.5. So that would be, 100 100 to 150 or something like that nanomoles per liter. So
yeah, which according to national recommendations is too high, and I mean the minimum that they're looking at is pretty much like on a population study level. What would be the level where we don't see population-wide issues arising? So an optimal level, as optimal serum level for you doesn't necessarily mean when it comes from these population studies and national averages that it's optimal. It can be optimal from a national level to avoid a common issue, but not necessarily for good health.
So for this coming to this conclusion for 40 to 60 nanograms per mil, I've looked at around 33 different clinical studies that have been done from 1960s all the way to 2013 or 14. Looking at all-cause mortality and vitamin D, serum vitamin D levels. So all-cause mortality means people that die from cardiovascular disease, cancer, respiratory disease, all these diseases that people die from. And 40 to 60 nanograms per mil was associated in 33 different studies, with the lowest all-cause mortality. So that's sort of where I figured that's a sweet spot for the vitamin D, And also, again, we talked about this CYP2R1 gene polymorphism where people can't convert vitamin D3 into 25-hydroxy vitamin D very well. Those people with that gene polymorphism actually have a higher all-cause mortality than people without it, which is kind of an interesting way of looking. And they have lower serum vitamin D levels. So they have lower serum vitamin D levels, and they have a higher all-cause mortality. So
right on. So let's touch a little bit more on longevity. And I mean, in Finland, here in the north, today was the first snow that came. I'm super excited. I'm also excited of winter because of the cold exposure that I can have on all the heat stress that I can accumulate in going to the sauna every day. I'm actually doing that yesterday. I'm probably going to do it tonight as well. So heat stress, also a hormetic stressor, just like some food nutrients can be, mild toxins, but at the same time they are making our immune systems better. So what can you tell us about heat and its effect on longevity and especially cold?
Well, I do. I kind of want to start maybe with
it's a complex issue.
If we go back to my longevity worm study, you remember when I first started studying aging. So that the heat goes back to that because back when I was studying these little worms, we would heat shock them. We'd expose them to a very brief burst of heat and see how that affects their lifespan. And
so you had little worm saunas they would go in and throw a little water on the stones and enjoy themselves.
Yeah, well, not quite that much, but it was kind of like a little worm sauna where you just kind of expose them to like a 15-minute, heat shock. And this has been done by several different laboratories. In fact, it's been published by another laboratory, not the one I was in, but we were doing it to repeat the experiment and to look at other things. And what we found was that heat shocking these worms just for a very short, brief, it was 15 or 30 minutes. I don't remember, it was so long ago, but it extended their lifespan by like 20%, 15 or 20%. Which isn't quite as robust as the IGF-1, but still just one single exposure to heat, So I've always in the back of my mind, connected the heat exposure to longevity just from my early, exposure to the literature on heat stress and how it was able to increase longevity in these lower organisms like worms and also flies has been shown the same thing. So I've sort of always been in the back of my head, how there's some mechanisms and a lot of those mechanisms were showed to be dependent on activation of a pathway called heat shock, the heat shock response, which activates heat shock proteins. Heat shock proteins are they're proteins that are doing a lot of things, but one of their main functions is to make sure that a protein has its proper three-dimensional shape and structure so that it's able to do its function when it's supposed to function, and also so that it doesn't, after it does its function, it is degraded normally and it doesn't stick around with an abnormal structure where it can then aggregate with other proteins. So heat shock proteins play a very important role in preventing protein aggregation. And protein aggregation plays a role in a variety of age-related diseases. It plays a role in Alzheimer's disease, Parkinson's disease, Huntington's disease. It plays a role in atherosclerosis and cardiovascular disease. So proteins will aggregate in your blood vessels, and you can start to have all sorts of nasty stuff just sort of lining the blood vessels, forming a big aggregated, plaque. So heat shock proteins are one of the interesting hormetic stress response pathways that are activated when animals and when humans are exposed to heat. Humans also, when they're exposed to heat, activate this heat shock response. But I think probably one of the most convincing studies. So these studies, there have been studies done on worms, flies, and even mice, where you heat shock them, and you can improve the way they age. You can extend their lifespan by 15% or so. But there was a study that was published, I believe it was in 2015, and it was a study that came out of Finland, the University of Eastern Finland. And the study looked at all-cause mortality, so dying from cardiovascular-related diseases, cancer, all sorts of diseases, and sauna use, frequency of sauna use. And what the study found was, so it looked at men that use the sauna either once a week, men that use the sauna 2 to 3 times a week, or men that use the sauna 4 to 7 times a week. So it's kind of like a range of using the sauna, like a dose response in a sense, where some men are using it more frequently and some are only using it one time a week. And what the study found was that men that use the sauna 2 to 3 times a week had a 24% lower all-cause mortality than men that used it 1 time a week. So they actually died, they were 24% less likely to die of all these different diseases during the time frame that was studied.
Right.
Men that used one. And then
there is so many benefits of that. Also, the increase of white blood cells.
Yes.
When you go to sauna as a kind of a hormetic stressor, so reduce the risk for common flu. Also, the way how it shuts down inflammation. I just heard a pretty interesting story I want to share with you. I was in the Finnish Sauna Society with one of the top personal trainers of Finland called Tomi Kokko, and he told he used to go to this and still goes to this place where there is smoke sauna, and there's this one guy who's always there, and he would be in the ice water, and basically there would be ice, and he would dig himself a hole. And Tommy would go to sauna and outside and swimming and sauna. He would do this like 3 times. And the guy is still there in the ice cold water. So he was like thinking of calling an ambulance because that guy looked like hypothermic and he was just like lying in the ice water. And then he struck a conversation with that guy when he came out of there. He explained that he's basically getting himself to controlled hypothermia intentionally, because that's the only moment when his osteoarthritis symptoms go away and the pain goes away, and much better than any medication. So he does that treatment to himself every day, like going to 20 to 30 minutes to ice cold water to control hypothermia. He looks like dying, but he's kind of like, he knows what he's doing, and he's just like lying there in the water. So what would you say about the kind of immediate kind of effects of just like getting into the ice cold water?
Into the ice cold water. Yeah, that's a little different. That's a different type of stress, but one of the most irrefutable pieces of evidence that I've seen in the scientific literature is that cold exposure immediately increases norepinephrine, which is it's a neurotransmitter, but it's also a hormone. So norepinephrine, the reason it increases norepinephrine is because norepinephrine causes vasoconstriction. So when you're cold, you actually are trying to conserve heat. And the one way you conserve heat is by vasoconstriction. But in addition to that, it has norepinephrine when it's in the bloodstream. It's an anti-inflammatory. It actually inhibits a variety of inflammation pathways, TNF-alpha, NF-kappa B. So it is a very potent anti-inflammatory. So it has its role in inflammation. It also affects immune cell function, white blood cell number, and it also affects mitochondria. So it's playing a role in causing your mitochondria to uncouple. So it plays a role in uncoupling mitochondria, which means the mitochondria are no longer coupling oxygen to make energy, and they kind of freak out. And as a response to that, it's sort of like a hormetic stressor. They start to become very active and try to make more and more energy to generate heat. But then what ends up happening is it also causes you to make more mitochondria when this pathway gets activated, because your body's like freaking out because it's trying to warm you up. So it ends up having this net positive effect of where now you start to make more mitochondria.
So you will have better, you will have more energy, like long
you will
term.
have more energy. And then the norepinephrine in the brain also gets activated, and that plays a very important role in making you focus and focus and attention are better. It's also an antidepressant, so it makes you feel good. And as you mentioned, in pain, it's not quite understood all the crosstalk, but it plays a role in somehow in alleviating some of the pain pathways, and so it's very interesting how it's sort of having all these pleiotropic effects where it's affecting so many different physiological processes.
Right. What can you tell about the genetic side? I remember you raving a lot about a gene called FOXO3 that's linked to this.
Yeah, FOXO3. So that's going back to the heat stress. And I was talking about the study out of Eastern Finland. The all-cause mortality was even lower. It was 40% lower in men that used it 4 to 7 times a week. A lot of that mortality was actually related to cardiovascular mortality. They were less likely to die from a heart attack, heart disease, coronary artery disease. And that has a lot to do with the fact that heat stress mimics cardiovascular exercise to some degree, and your heart rate elevates. I mean, anyone that's sat in the sauna knows that it almost feels like you're doing an aerobic workout. So that there's a lot of similar effects that are activated. Anyways, that's just the cardiovascular side. The other thing, and this has sort of not been proven so much as me sort of connecting the dots, and that is that heat stress, because it is a hormetic stressor, it has been shown to actually activate the longevity pathway called FOXO3. And this FOXO3 pathway is it's actually a very important stress response pathway that activates, many, many different genes that are involved in dealing with stress, and antioxidant, anti-inflammatory, stem cell production, just clearing away, damaged cells through autophagy. It's just very, very good pathway. And in fact, humans that have a gene polymorphism in their FOXO3 gene that makes it active all the time, makes it more active, they're 2.7 times more likely to live to be 100 years old, so a centenarian. And if you look in animal studies, for example, in mice, if you activate FOXO3 all the time, the mice can live up to 50% longer. And also this worm study that I first talked about when we started the podcast, part of the reason why the worms lived 100% longer was because getting rid of that insulin growth factor pathway, it activated FOXO3 all the time. And so if you got rid of the FOXO3 also, that lifespan extension was gone. So it required that FOXO3 pathway, which worms actually have a version of. It's a little different, but it's the same gene, essentially.
Just a quick question. So there's studies that show that cold exposure, for example, after exercise blunts growth. I mean, if you want to put some muscle or increase strength, doing cold exposure right after exercise is not necessarily good. Is it also linked to this reduction of IGF-1 or increase of FOXO3 when you're kind of, or is it just like shutting down the inflammation and damage?
Well, actually both. So it is, doing cold exposure like an ice bath immediately, like within 1 hour after doing strength training, resistance training types of exercises, can blunt the hypertrophy, so muscle growth that is experienced from doing the strength training. And that has been shown to be, to some degree, dependent on stopping inflammation, which is important for activating the IGF-1 in muscle cells. So it's kind of both IGF-1 and the inflammation, because the inflammation that you generate from, engaging in physical exercise, from strength training, is very important. It's the hormetic effect, right? Exercise is also a hormetic stressor, much like heat and cold. And so if when you exercise, you're actually increasing inflammation, and that increase in inflammation is essential to have the response, the anti-inflammatory response to the inflammation. It's even more powerful. It's more robust. So, and also that inflammation is important to activate the IGF-1 in muscle. So without it, if you're blunting it too quickly, you're not going to have that very important response. So it's important to keep in mind that, at least in the context of strength training, that it's been shown, if you do it 1 hour, so if you wait 1 hour, after 1 hour, the inflammatory response has already reached its peak, and now you're already in the anti-inflammatory response mode. You're already having that stress response, that hormetic stress response to the inflammation. I think this is what I've seen studies that have waited an hour and it doesn't have that same effect. We still need more empirical evidence to actually prove that, but it is my belief that waiting at least 1 hour after strength training to do cold exposure would be more beneficial than. Now, aerobic exercise has been a little different. It hasn't really been, in fact, it's been shown to improve endurance if doing it immediately after endurance.
So there might be a benefit of doing cold exposure if you want to be recovering faster from a strenuous exercise day, and you need to do second round the next day. Now heat, like sauna after strength training, compared to like stretching. Actually, you don't need to stretch after strength training to reduce muscle pain. You just go to sauna, enjoy yourself, and that's often seems to be more effective than doing all the stretching and using your time on that. And on the top of that, with heat shock proteins and that activation, you get some longevity benefits, health benefits of that as well. So very cool. Thank you so much, Dr. Rhonda Patrick, on this advice. I think we're coming close to our end of our interview. But if you would pick up from your research on heat and nutrition a few kind of like main takeaways that people should do more research on their own, which ones would you pick? So what should they go and look for, and where should they go and find that information?
Well, I think that the vitamin D is a very important one, and getting your vitamin D levels measured is important. I have a video out there called the Vitamin D Sweet Spot, which is a very short video talking about the role of vitamin D in longevity and what I have, in my opinion, I have identified to be the important blood levels of vitamin D, the range to be within. Also, we talked about nutrigenomics. I also put out a video on that, and I have a free nutrigenomics tool that can be found on my website, foundmyfitness.com, where you can learn about a variety of different gene polymorphisms that affect vitamin D, that affect folate, that affect vitamin A, vitamin B, that affect omega-3 fatty acids, the way your body's converting them. Also, that affects fat metabolism, how you metabolize fatty acids. So that video is on my website. It's the nutrigenomics video, and also there's a free tool. If you've done a 23andMe genetic test, then you can run it through my website, and it'll give you some information on some of the genes that I've identified. I have some more genes I'll be putting out, more reports I'll be putting out soon. So I think that's another important take home. And then the sauna. I know the sauna is really big in Finland. I think some of the really important take homes with the sauna.
We have more saunas than people, and every Finnish person is a sauna expert, by the way.
Well, we didn't actually get into the effects on the brain, which is what I'm going to be talking a lot. I'm going to be talking a lot about the sauna at the Biohacker Summit coming up soon. And it actually was one of the most important things that got me interested in the sauna in graduate school was the effects on the brain. So it has a very profound effect on the way your brain responds to endorphins, which makes you then feel good even days after the sauna. So that's something that's very interesting. But the stuff that we talked about today, the heat shock response, heat shock proteins, the FOXO3, the effects on the cardiovascular system, I have a video on that that's called the sauna longevity, the effects of the sauna on longevity. I also have a free report you can find. So if you're someone that likes to read, you can download those free reports on my website. I also have one on cold shock. I put out a podcast on it, an audio-only podcast, which can be found on iTunes, Stitcher. It's called Sound My Fitness. But I also have a free report on 20 pages, and it's on cold shock, and it's how the many effects of cold shock on inflammation, on the brain, on making new mitochondria, mitochondrial biogenesis, on metabolism. So that can also be found on my website. But
yeah, that's it's a very cool article. I will link all of these into the show notes. And foundmyfitness.com would be the address, and also Found My Fitness on iTunes and YouTube channels. So check it out. Dr. Rhonda Patrick has a wealth of information you definitely want to dive deeper into that we can't cover in this podcast episode. So thank you so much, Dr. Rhonda Patrick, for your time, and also for coming over to Finland and link up with all the crazy people who do the sauna and cold exposure every day and in the pursuit to live longer. Unfortunately, our region is not in the blue zones. I guess it's because of the lack of sunshine and lack of diversity in food. But at least we mitigate a little bit of the damage by just like getting ourselves out there into the nature and getting our little worm kind of genes activated for longevity. So very cool. Thank you so much for your time.
My pleasure, and thank you so much for having me. And I'm very excited to come to Finland and experience the saunas and ice lake or ice cold shock after. So I look forward.
Absolutely. So everyone out there, so have a healthy week and stay upgraded. Thank you very much. My name is Teemu Arana, and check biohackersummit.com for more on the conference. Dr. Rhonda Patrick is going to be there. Cool. All right. Over and out. Bye.